Welcome. I'm Dr. Ruff, and we're here to talk about the latest advances in thrombosis prevention, including the emerging factor XI inhibitors. Before we begin, though, I'd like to thank our audience for joining from all the different social media platforms. This is certainly an exciting topic, so please do submit questions as we go, and we'll address what we can in the time that we have. So I'm pleasure to be joined today by my friend and colleague, Dr. Michael Gibson. Great to be here with you. We're fresh back from the London European Society of Cardiology Congress. And what were some of the key updates and takeaways in thrombosis? I know you were the chair of a couple of the AFib hotline sessions, and we'll talk about some of them together today.
Well, thanks, Christian. It was an exciting meeting. There were updates about the ability to implement guidelines. We do a better job in AF when we do some education in terms of arrhythmic control. The other thing in the AF session that I chaired was a study looking at edoxaban monotherapy in those people who have AF who got a stent, and it was as good as combining it with an antiplatelet. But the real highlight of the meeting was Oceanic AF, the first big trial looking at a factor XI inhibitor in the setting of AF. And that's, I think, going to be a great discussion today.
Great. We're definitely going to do a deep dive of that one and the implications for the field. But before we get to those, I thought it might be helpful for our audience to really set the stage how we even got here. Why do we need more anticoagulants? What's the matter with the ones that we have? Well, fortunately, we do have very good anticoagulants, but there really remains a significant unmet need. We know that there is pervasive undertreatment. That's generally driven by a fear of bleeding risk or patients who have bled before. And we know many patients often don't even receive an anticoagulant, or when they do, they frequently discontinue, either because they've bled or they're worried about bleeding. And the number who are undertreated varies, but it could be as high as 40 to 50%. And so that's really striking. Sometimes even patients who are getting our anticoagulants, particularly the DOACs, which are our current sort of platform of anticoagulation, many of them aren't getting the correct dose. They're often underdosed. And that's really driven by this fear of bleeding. Unfortunately, when you underdose, those drugs are less effective. So this is a significant problem. Patients aren't being prescribed it, they're taking the wrong dose, and they're frequently missing doses or taking it incorrectly. So we really do have a bridge where we need a better anticoagulant for our patients. And there's a lot of factors that kind of drive this undertreatment. Providers are concerned, the sin of omission versus commission. They don't want to prescribe a drug that causes bleeding. And we know these patients are generally frail, they're elderly, they have a lot of comorbidities, they're on a bunch of medications. Patients obviously don't want to have a stroke, and we'll talk about some of the other indications, MI, et cetera, but they're often worried about bleeding. If you prevent clotting, don't you cause bleeding? And obviously, there's some confusion about the dosing. And then obviously, there are a lot of other issues that go into sort of chronic administration of these medications. Cost and access is always an issue that we have to tackle. And clearly, there's some opportunities for us to improve on our current level of anticoagulation. And so, with all of that, we've come to the promise, really, the holy grail of trying to prevent clotting without causing unnecessary bleeding. And there's the promise of factor XI pathway inhibitors, and there's certainly a lot of excitement in this field. And so, Michael, can you sort of orient us to, like, why is factor XI inhibition such an intriguing target, and how is that different from our current anticoagulants, such as the DOACs, the factor Xa or 2A inhibitors?
Well, when I mentioned we were doing research in this field, my son just kind of rolled his eyes in the back of his head. Why are you doing another big trial? Nature's already done the randomized study. And in 2 minutes, he got on the UK Biobank and he said, you're right. People with factor XI deficiency have lower VTE event rates and CV event rates, and they aren't having a lot of bleeding. That's the hope. Now, on this slide, you see how we're separating out the two pathways. Now, this is a bit of an oversimplification for you hematologists out there. I get it. But on the left-hand side, you see the extrinsic pathway. Extrinsic, meaning outside. These are the clots we want. These are the clots that prevent you from bleeding to death. They stop bleeding. On the right side, those are the clots we don't want. The intrinsic pathway, inside the vessel pathway. Those are the clots that form in your artery, clog it off, and cause a heart attack or stroke. Here's the key. If you kick off the left-hand side or the right-hand side, you got to go through factor 10. By blocking that, it doesn't matter how you activated the system, you're going to get both efficacy and a safety problem. Now, look over on the right-hand side. If we just block factor 11, then we can block that side of the cascade selectively and preserve the good clotting on the left-hand side. So allow good clots to continue to form that stop bleeding, but prevent the bad clots from forming on the inside of the vessel. That's the hope. I think we've seen some good safety so far.
That's terrific. So I think I got it. Factor 11, the promise is we can form the good clots, repair injured blood vessels, protect ourselves from harmful bleeding. But the bad clots, the clotting in an artery that causes stroke, or in the veins, venous thromboembolism, or say MI also in the arteries, we prevent those clots. And as you said, there's been tremendous enthusiasm for this pathway. There are lots of different approaches. There are antibodies being developed to factor 11. There's antisense oligonucleotides, which prevent the factor protein from being generated. There are small molecules that bind to activated factor 11. That's the part that does the dirty business of clotting. Some target both factor 11, which is the inactive precursor in the liver, other the active version. And the fascinating thing is they're being studied across a whole range of conditions, including cancer and end-stage renal disease. We'll talk a lot about atrial fibrillation, but let's get sort of a broad brushstroke and say, what have we learned from the phase 2 data from these studies, which were done, predominantly in preventing venous thromboembolism in the orthopedic population. We have trials in myocardial infarction, and obviously the stroke, mostly atrial fibrillation, but from secondary stroke. And I think first, maybe I'll start with the venous thromboembolism. That has really been the proving grounds of anticoagulants that show us that this mechanism prevents clots. And many of the agents were studied in preventing clots in the venous system, a DVT or PE, after total knee arthroplasty. And we know that several of these agents were highly effective in reducing clots on the venous side, which is terrific. That has usually translated also to preventing clots on the arterial side. So again, proof that this mechanism works, preventing clots. There were also some very important studies in phase 2 for secondary prevention for non-cardioembolic stroke, so stroke that's not due to atrial fibrillation, as well as in patients with an MI. And that's really important because that's a different question. Can we use an anticoagulant safely in combination with antiplatelet therapy? Because we know the cornerstone of therapy after an MI or after a stroke that's not due to AF is antiplatelets. And so we know adding an anticoagulant to that might be effective, but the bleeding is just intolerable, even with the DOAC. So if we had a super safe anticoagulant with the factor 11 inhibitors, could we use combination antithrombotic therapy? Now, at least in the phase 2 studies, again, not powered for efficacy, so we don't know if they're effective on the arterial side, but it appeared to be very safe to use in combination with either aspirin or one of the P2Y12 inhibitors, or even both. And then obviously we have two, I think, important phase 2 studies in atrial fibrillation that I'd just like to spend a little bit more time on. That's the Pacific AF study. Again, that was with one of the small molecule factor 11A inhibitors, asundexian, and that showed, compared to apixaban, and again, a small study, only a couple of months in duration, that there was a reduction in bleeding compared to apixaban, again, underscoring the safety of this agent. About in the pooled analysis of asundexian, a 2/3 reduction in bleeding against apixaban, which is thought to be our safest anticoagulant. The safety, I think, of the factor 11 hypothesis was really confirmed in the larger Azalea study, which we led at the TIMI study group, which demonstrated marked reductions in bleeding with a monoclonal antibody to factor 11, abelacimab, which reduced bleeding compared to rivaroxaban by around 70 to 80%. And that included major and clinically relevant non-major bleeding, that's really clinically important bleeding, as well as major bleeding alone, almost eliminating gastrointestinal bleeding, which is remarkable because that's about 2/3 of the bleeding we see in atrial fibrillation population. And again, this incredible safety was observed even though abelacimab is extremely potent. It reduces factor 11 levels by 99%. And so a highly potent inhibition of factor 11 is incredibly safe. And I think people were really excited then for the potential. What does this mean? Now, of course, and Mike, you and I go way back as clinical trialists. Everyone always says the caveat, you have to wait for the phase 3 trials to show that these drugs are effective. And right after the AHA, when we presented Azalea, we heard the surprising and disappointing news that the first large phase 3 trial in atrial fibrillation, Oceanic AF, with asundexian, the same agent that was used in that earlier Pacific AF study, was terminated early. The trial was stopped by the Data Monitoring Committee for inferior efficacy compared to apixaban. And so we sort of held our breath for several months. But that was just recently presented and subsequently published in the New England Journal of Medicine at the European Society of Cardiology Congress that we were at. You chaired that session. So Mike, what happened? Tell us what your thoughts are on Oceanic AF.
Well, first of all, none of us know for sure, but I think, as I said in the session, you have to give the right drug at the right dose to the right patient. Let's take those three things. Right drug. Do we think the hypothesis is right? I think the consensus view is yes, the hypothesis still remains well-founded on the venous system, 4 trials, 4 drugs, all showing a reduction in clotting and a reduction in bleeding versus enoxaparin. This is an active biologic target. It works in the venous system. Now, is the atrium more venous-like or arterial-like? Seems to be a little more venous-like.
Right.
So I think most people think this is a viable biologic target. Number 2, right dose. The consensus view is that the asundexian dose was underdosed. Now, we all know that if you give once-a-day dosing, you give high peaks and lower troughs. If you give twice-a-day dosing, you get lower peaks and higher troughs.
Right.
So asundexian was once a day, had higher peaks, but also had probably lower troughs. Milvexian, given twice a day, had lower peaks, but higher troughs. To put some numbers on that, the trough level for 50 of asundexian once a day is about 1.6 times the upper limit of normal. For milvexian, it's about 2.5 times the upper limit of normal.
Right.
So higher.
That's the other factor 11 and the factor 11
small
inhibitor.
molecule.
Yeah.
Now there's a third molecule, abelacimab. This is a sub-Q injection given once a month. And imagine this, you don't have peaks and troughs. So that issue kind of goes away. The other issue is the magnitude of factor 11 inhibition. There was an internal assay developed by Bayer that seemed to indicate good inhibition with that assay, but standard assays indicate that the inhibition may not have been as good as we thought. As you mentioned earlier, the real leader here in terms of potency is abelacimab with 99% inhibition. Now, if I had to have a factor deficiency, I'd want it to be factor 11 deficiency.
Right.
When you look at major bleeding, you really don't see major bleeding, very infrequent with factor 11 deficiency. So it should be safe. And when you look at the totality of the phase 2 data, this phase 3 data, it is safe. Now, the other issue is the right patient issue.
Right.
Those people who were naive to factor or to oral anticoagulant, they tended to fare better with asundexian. But none of us have really been able to understand that subgroup. It's small numbers of patients. Strangely enough, the naive patients who received asundexian had a dramatically lower event rate than those who were experienced with oral anticoagulation, which doesn't make
Doesn't
sense.
make sense intuitively.
So many of us don't really believe the subgroup analysis. So at the end of the day, the consensus view is the hypothesis is viable. Numerous studies are ongoing, pretty far into the development program, have not been stopped for a signal like we saw in Oceanic. I think the venous system is very clearly a winner, and we've just got to let these trials run out to see what happens.
No, great summary, Mike. And as you mentioned, Osindexin was one of the assets that did not show efficacy in the VT. They did not do a VT study, so we didn't have evidence that that drug at that dose reduced clot in a clinical setting, where these other agents, as you said, in late stage phase 3 trials did. And we know they're more potent too. They knock out factor 11 pathway with much higher potency than
Well, they're
others.
much more potent, like abelacimab, but look at
Right?
the dramatic reduction in bleeding that you saw with abelacimab versus rivaroxaban. So yes, very potent, but also
Yeah.
very safe.
And
Yeah.
I think to add on to your comment about the remarkable safety at ESC, my colleague Sid Patel from our group presented the peri-procedural data out of Azalea, and what was once unthinkable, a long-acting anticoagulant, dosed monthly, inhibits factor 11 by 99%. What happens to patients needing a procedure? As you know, with rivaroxaban, apixaban, the other DOACs, we're starting and stopping these medicines all the time, holding it for many procedures. What did we learn from a long-acting agent? Is that something safe to have? And we know a lot of patients had procedures in the Azalea study. We know that there was a whole range of procedures, and many of the procedures are low risk procedures, but we did have high bleeding risk procedures and intermediate bleeding risk procedures. And remarkably, over half of the patients in the abelacimab group who had a procedure had it within the monthly dosing interval. So we're fully therapeutic on abelacimab. So 99%, reduced levels of factor 11 when they had the procedure. And remarkably, they were actually numerically, bleeding was very low. There were actually numerically less bleeds in the abelacimab arms compared to rivaroxaban, and that's despite rivaroxaban routinely being discontinued entirely before the procedure. So that most of those patients aren't on any rivaroxaban. And so I think it's like you, it's a real paradigm shift that it's so safe, these factor 11 inhibitors, if their promise of efficacy ends up being fulfilled, that we may be able to provide effective anticoagulation with a remarkably safe bleeding profile that we've not even gotten close to historically in the last 60 years of oral anticoagulation. And so, but obviously, as you mentioned, we have to stay tuned for the ongoing phase 3 trials, because these other agents, I still think, and echo your comments, that have a lot of hope. We know that antibody to factor 11 abelacimab is in phase 3 trials as well. It's in another AF trial, the Lilac TIMI 76 trial. This is a very high risk AF population, patients ineligible for currently available anticoagulants. So these are very old, frail patients, the patients most in need of a safe anticoagulant. And then 2 cancer-associated VT studies, really high-risk patients. They already have a clot. Cancer, we know, drives thrombosis on the venous side. And so we wait for those Aster and Magnolia studies to finish. You mentioned Osindexin, no longer Oceanic AF, but they are doing a secondary stroke prevention trial that's ongoing, the Oceanic stroke trial. And then you mentioned the real promise of milvexian, the other factor 11 inhibitor, a real constellation of trials, the big Librexia AF study being done against apixaban, of kind of a very similar trial population to Oceanic AF. So we're really excited about that, but also being studied in secondary stroke and Librexia stroke, as well as a trial I know that you're co-leading, Librexia ACS. And so we'll really get a huge amount of information, atrial fibrillation, secondary stroke, after ACS. And so we're really excited about the potential of these agents. And I think stay tuned, and hopefully we'll have one of these sessions when these other trials start to report. And so, where do we go from here? What are the real world implications? Obviously these drugs aren't approved yet, so it's still the promise. But what do we know? We know that these drugs are remarkably safer compared with DOACs in phase 2 trials. That is certain. That part of the hypothesis is held up. Factor 11 inhibition is remarkably safe compared to DOACs. And that's really, I think, an intriguing option potentially for patients. I do think that Oceanic AF like you was disappointing, but there's a lot of emerging data that there's significant differential potency in this class. We didn't really see that in the DOACs, but it appears to be the case here. They have different mechanisms. They're dosed differently. The potency is very different. So we really have to take each trial and each agent at its own merit. And I think you really touched on these drugs are being studied in different patient populations. Factor 11 inhibition, dial up or dial down in different scenarios, depending on if it's post ACS, secondary stroke AF, is it in combination with antithrombotic therapy such as antiplatelet therapy? And I think I share your optimism that we're likely to see significant efficacy in phase 3 trials. And what would your key takeaways, Mike? You and I have been doing this a long time, and you're always so insightful. What would you like to tell our audience as far as, what are the things they should remember about Factor 11 hypothesis?
You've all been randomized to a trial without your consent or knowledge. You've all been randomized to either normal Factor 11 levels or low levels. And the people with low levels, they don't really bleed, and they have much, much lower rate of thrombotic events. So nature's done the experiment, and it works. Clear, as you say, on the bleeding side. There really looks to be both phase 2 and phase 3 data now really showing that this approach is safe. The good news is that means we can push hard. We can push hard on the efficacy side. And, abelacimab at 99% inhibition is pushing the hardest. Milvexian's probably pushing the second hardest in terms of inhibition. We got to wait to see what happens in these phase 3 studies to know whether this is going to be effective. They have not been stopped after large numbers of patients being enrolled. So let's wait till the end. I'm optimistic. If it does work, for the first time in 40 or 50 years, we may get a free lunch. To this point, the free lunch has always come with a little ketchup.
Right.
Bleeding.
That's right.
First time, we may get a free lunch without ketchup. So let's see what happens.
Oh, fantastic. A great summary. And I think this is our favorite part of the program is obviously to take some questions from our audience. We already have some great questions. I'll start off. I think, someone asked, how do you abort Factor 11 action when needed for interventions? I think you mentioned the experiment. We learned about Factor 11 deficiency. There are patients walking around with no Factor 11 at all. And fascinatingly, unlike the other Factor 11 deficiencies, which are the hemophilia patients, these patients don't bleed spontaneously. They don't seem to bleed even with major procedures for the most part. And I think the Azalea periprocedural data show us that you could have less than 1% Factor 11 and you're undergoing all sorts of procedures. And so the thought is you may not need to abort Factor 11 action for procedures. Now, we'll learn more from the larger phase 3 studies, get more experience in high bleeding risk scenarios. But I think our whole thinking about, we got to be scared of them being on anticoagulant. For every procedure, we got to stop, and then restart. That might not be necessary. Like when you have a procedure, you don't stop your statin, right? There's no reason to stop most of your medications. Maybe if anticoagulation is so safe, we can have that similar approach. So that's the promise. Obviously, more work to be done. I think, one of the questions that comes up a lot for me, Mike, I'd like to ask your opinion is, for obviously a lot of appeal for Factor 11 inhibitors, but the DOACs are omnipresent, right? They're going to be generic or are generic depending on where you are. Who will be getting these Factor 11 inhibitors? Who should get them if they're approved based on the phase 3 trials? Do you think everybody or just a subset of patients? What's your thoughts?
Well, this is the way it always works in drug development. You often show that it works for everybody. And then you've got to figure out, well, who would I start with? If I was going to start to use this new drug. And, it's those people who are probably on the edge of both efficacy and safety. Those tend to run together. The highest risk bleeders are often the people who are at highest risk of bleeding. So I would say the high risk bleeders, those would be the people who I'd start with. If they require anticoagulation and you're concerned about safety, this would be an excellent group to start with. One thing we didn't get to mention today was some of the safety data looking at people who had prior microhemorrhages
Right.
will
Mhm.
increase harm with this group. That's something that makes people fearful. And also no increase in microhemorrhages in the brain with this factor 11 group. So I think we'll see people start with the bleeders and grow from there.
Thanks, Mike. And what do you think? There are so many, all the DOACs were they're all oral, small molecules that kind of worked in a similar way. Here, there are antibodies to factor 11. There are small molecules to activated factor 11. There are other antibodies to both factor 11 and activated factor 11. There's ASOs, there's going to be siRNAs. It's sort of like a mechanism jumble. Do you think there's any inherent advantage of one mechanism for the other? Do you think it's going to all boil down to potency?
Well, if you want it to work, it's got to be potent.
Right,
If you want a patient to take it and be adherent, then it's got to be convenient. I
right.
take a couple of injectables. I get them in the mail, refrigerated, once a week, once a month, whatever. I sit in my home and I take them. I'm compliant. So I do think with this kind of sub-Q approach, adherence might be very good. Tolerance might be quite good. The other limitation is always looking at interactions between drugs.
Right, very
So
important.
the ones that play well with others will predominate. The ones that are most convenient will predominate, and the ones that improve adherence will predominate if potency is similar.
Oh, that's terrific. And a lot of, one of the things that also comes up, and I know you spent a lot of time thinking about combination antithrombotic therapy. And there's other data, as you mentioned, for the edoxaban data. You mentioned the EPIC CAD. Which seems like we're on this journey of pulling off as much antithrombotic therapy as possible in post ACS or certainly in stable CAD. You know, how will the potential data for factor 11 inhibitors work? Is that maybe the wrong idea? It's just that, we couldn't tolerate combination antithrombotic therapy. Now we're really talking about maybe we should be adding on an anticoagulant. What are your thoughts? Do we need more efficacy? Somehow that seems counter to what we're all doing, which is like, let's keep taking away an antithrombotic. Maybe that's not the right thing.
Well, I'm an interventional cardiologist and I practice in that field. And I know we've been taking things away, particularly aspirin. I
Right,
mean, aspirin, I think, is now in the ESC guidelines. They now say clopidogrel is a suitable alternative to aspirin
right,
monotherapy. So we're peeling away aspirin, a lot of gastric bleeding. Uh, you know, there's a patient attached to the stent, and that patient needs secondary prevention.
right.
And what we have seen, 5 trials now, Atlas 1, Atlas 2, Compass, for Paxar, warfarin. When you inhibit thrombin generation in the ACS setting, you improve efficacy. And we saw reductions in mortality in these studies.
Amazing. Yeah.
People weren't willing to use them because of the risk of bleeding. So, if we have a strategy that is safe, but it is also effective, I think you'll see a resurgence in use in this class, this approach to inhibiting thrombin generation. I think one of the centers of bullseye no one's studying, but I want to study it, is the VAD patient.
Yes.
We have good treatments that prevent thrombosis. That's good. We have really a big risk of bleeding. 40% of these people will bleed. And, right now they're on warfarin. We dropped aspirin. They're still bleeding. So I think that's an area, the VAD patients are still a group of people looking for a safe alternative. Everywhere we see a need for safe alternatives, I think that's where this group's going to shine.
No, it's terrific, and I think that's a lot of the excitement. I know in addition to potentially the VADs, maybe even when we visit mechanical valves, a lot of interest in end-stage renal disease as well. High thrombotic risk, high bleeding risk, dialysis catheters, clotting. And so I think there's a lot of opportunity to factor 11 inhibitors to really move the needle and kind of protect patients across a whole range of venous and arterial thrombosis. And so I think that that's really an important message is that, now that we have an incredibly safe anticoagulant, we're seeing bleeding that's similar to prophylactic low molecular weight heparin with full dose anticoagulation. And so I think if the promise of efficacy prevails, that's really going to be a game changer. And I'm excited to be on that journey with you and our audience. And it was a pleasure talking with you, getting your expertise today, Mike. And I'd like to thank our audience for participating in this activity.
Thanks, Christian.